Perfect. So we'll go ahead and get started, everybody. My name is George Magrath. I'm the CEO at Lexitas, and we're really uh, grateful and thankful to have Dr. Tauber here. And Dr. Tauber, do you want to give your introduction?
Sure. You want to make an attempt at describing me, or you want me to do a self-present?
so, so Dr. Tauber, I'll give an attempt to describe him, but Dr. Tauber is a world's expert in dry eye development, right? He has been doing this for a long time, and with Lexitas for a long time, and has been a part of many, many development programs, and is one of our go-to KOLs for dry eye disease.
Well, so we've
Great.
established that I'm old. That's fair. So I've been in clinical practice, some 30 years, cornea specialty, ocular immunology specialty, and I started doing clinical trials way back then, really in my fellowship. I actually recall it as the first cyclosporin study. Can't say Restasis because that was before Allergan even purchased it, and I developed an appetite for it, and I've never stopped doing clinical trials. So I think I'm over 150 or so, and of course, you do a lot of trials, you wind up on advisory boards and medical boards, and you wind up hearing what happens behind the curtain. And having done plenty of clinical medicine, I feel like I've extracted all the gratification that I want from that. And I really want to shift to the other side. I want to move more to helping on the development side, because after 150, you see things that work well in studies. But unfortunately, you see a lot of things that go wrong in studies, and I want to cut down on that. So that's really what drives me. This is nothing resembling retirement, just to make more of a career shift into the industry side. And it's true, I've worked with Lexitas for decades, and I'm very impressed with how they do things, and I'm happy to be involved, very happy to join.
Yeah, yeah, and that's perfect, and that's exactly why we're here today, is to really talk about development in dry eye and start to talk about just a little bit about the past, and then get into sort of the present, and then a little bit about the future. And so, Joe, so sort of the first question I would pose to you is, how have you seen things shift in anterior segment over time as trials have been going on?
A whole lot. I mean, if you think about what's evolved in dry eye over these decades, it's unbelievable. But in terms of studies, it's all about convincing the FDA that you've got data sufficient to approve a new product. The classic was one symptom, one sign. Even that requirement has evolved quite a bit to where now you have co-primary variables in studies. So companies are doing 4 trials instead of 2. You need to show replication, so 2 trials that show symptom, 2 trials that show sign, as opposed to you must show them all together. And then you dig in a little further and look at the individual metrics that are being studied, and that's changed radically just in my time. Years ago, symptoms in dry eye, we had the classic 7 symptoms: dry, gritty, sandy, burny, foreign body sensation, itching, discharge, and blur. And those were graded on a variety of scales: mild, moderate, severe, some 0 to 100, very little uniformity. That's really gone. I think OSDI was used for a while as the symptom tool. That's not the best symptom tool for a lot of reasons. It's got a lot of functional assessments. So more and more, I'm seeing the eye dryness score really popularized. I think I should give credit to Shire for developing that one in the Lifitegrast development plan. Schirmer testing was very, very common. We all know the problems with Schirmer, and that's being used less and less. And I mean, as mean Schirmer. Allergan was the first to use responder concept, and that was really a post-study massaging the data to get to that point. And I mean, reporting the percentage of patients that had a 10 millimeter or more increase in Schirmer. So that's the responder analysis. And obviously that succeeded. We got Restasis. Since then, more and more companies are defining their own responders on a variety of measures, including stain. It's not clear if the FDA is going to accept all of those, but again, I mentioned it as big, big changes in how we do studies. We used to do tear breakup time, stopwatch, a very poorly reproducible method, in my opinion. But now we've got more machine-driven, non-invasive breakup time assessments. Exactly how reproducible those are is yet to be proven, but certainly better than a stopwatch, which includes reaction time and a lot of other things. how we record staining has evolved incredibly over the years, from the old Oxford system 0 to 5 to the NEI 0 to 15 to modified NEI, and now to the recently announced Lexitas corneal staining scheme that I imagine we'll get into a little bit later. Um, I think that's about all I would say on past. I think there are some future things, but maybe I'll save those comments till we get to future.
Sure, sure. Yeah. And one of the things you've always been talking to me about when I've asked you about development in dry eye has been the simplicity. And I think you hit on that, particularly with symptom assessments and going from where we used to have long questionnaires to more about just the VAS for dry eye and things like that, that are similar. Now you've had some experience with uh, with devices to help with the symptomatology. Is that correct? With smartwatches or iPhones or things like that? Do you mind sharing your experience with that?
Sure. I mean, I can share a bit. Some of that is proprietary, unfortunately, from other studies.
Yeah.
But again, it's all about how things have evolved.
Yeah.
There are wonderful symptom tools out there. But speaking as an investigator, and I think that's a perspective that
Mhm.
historically hasn't lived within a CRO, they're really tedious on patients. And when you have a patient sitting in the waiting room working on the 9-page questionnaire for 45 minutes, they don't like that. And even if they complete that study, they're not signing up for your next study. So tools like the VFQ 25, kind of lengthy. And more we're saying, how about you just collect what you need, not everything you would want. I think that's got a lot of advantages for the sponsor, because obviously you've got to pay for the collection, pay for the analysis, and then explain deviations that maybe you didn't expect. So I think, again, big picture, more and more trend to let's just collect what we need and let's not take more. There are people have been given iPads to take home and enter their symptoms. Yes, I've been in a study that used a smartwatch, which is a little challenging. I think 15 years from now we'll be in a different place, but I've still got a lot of dry eye patients whose watches are not as smart as they are, and they can't handle that kind of technology. Dry eye, we're really talking about the, let's call it 55 and above. I'm not going to call that older patients, because old is a word that changes like I do. But smart watches, where the instruction was every time you feel something, why don't you take your watch and let's input that, both the duration and the severity. And people can't stand that. Technologies are already so invasive in our lives. It's how deep do you want to get? And they, of course, like always, they go back and retrospectively enter things.
Mhm.
Any investigator knows, if you look in a patient's diary, you might see 14 days worth of information, but what do you know? It's all in the same pen. And they did it 10 minutes before they walked into your office. So we all want quality information, but the collection needs to be more and more efficient. And maybe we're moving there. I'm not sure that I love the smartwatch idea, but I think real-time collection is where we're going to go.
Yeah. I think it's I think it just highlights the operational challenges, the real world challenges of what should be a great endpoint to look at, which is the frequency and severity of exacerbations of your dry eye. Um, and so I hope it continues to evolve. I mean, it's exciting stuff, and hopefully we'll get there. I wanted to turn a little bit towards corneal fluorescein staining, if you don't mind, Joe, because this is probably the area that we spend the most time talking about from endpoints of dry eye studies, is exactly how to measure corneal fluorescein staining. And I know that I really love it that you have a pretty simple view of it that's very logical with using the reticle and the scale that you prefer. But can you walk us through how you think about corneal fluorescein staining and the different grading schemes and scales that are out there when you're thinking about trial design?
Sorry, your audio is getting a little choppy, but I think I understand the question. Unfortunately, experience comes from having made mistakes. So I've seen things evolve a lot over the years. Um, if you go back to Oxford, where you'd grade the entire cornea as a 0 through 4, in a sense, that was simple. It was a gestalt. I'm not sure I've ever seen reproducibility assessments, but it's not ideal. So then we said, well, let's break things up, because maybe you can be more specific or reproducible, at least if we did the cornea in 5 zones. And it's very logical. It's a central, I think it's 3 millimeters central and four kind of obliquely arranged areas around that. And the problem becomes really separating those areas. That got exacerbated when we moved from, let's not score from 0 to 3, let's start to count dots. Because where's the dot? Is it in the center, or is that in the nasal area? It gets a little bit subjective. And if you've got systems that involve, okay, the highest level is going to be up to, let's say, 38 dots. Just think about looking at a cornea, grading 5 areas, having to count up to 38 for each one. You're really going to lose some precision if you don't know exactly where those zones are. So, as you know, some Lexitas, I don't want to say sponsored, but managed trials used the reticle. That was wonderful. It's my favorite tool. I didn't want to give it back, and in fact, after I gave it back, I wanted to buy it back from you because it made life so easy to have an eyepiece that basically projected these 5 zones onto the cornea. It just made life so easy. So again, big trends. We've gone from Gestalt readings to counting dots. And then we got into the topic you and I have spoken about too many times, which is all the modifications to the classic
Mhm.
NEI system and debates about, well, is that really better? And is that really validated? And what does that even mean? What I'm referring to is we've gone from a 0 to 3, just vaguely, investigator impression, to counting dots. Well, do you use 0.5 units, or do you go with whole number units? Do you give a different weight to confluent spots? Do you give a different weight to macropunctate spots, which in my mind has always been rather difficult to define? What's a dot, and what's a dot that's significantly bigger? And then we've gotten into incorporating, well, what percentage of the area is involved with staining? I like to go back to basics. What is staining? And I think we don't even have uniform agreement on that, because I run into a lot of people who associate corneal staining with inflammation. And that's not really right. Staining is a demonstration of ineffective permeability barriers. It tells you the epithelium is not keeping out the vital dye. That's not directly associated with inflammation. And in particular, other pathophysiologic abnormalities can compromise permeability other than inflammation. So I think that's maybe helpful for someone to think about. So what are we assessing? How can someone with their eyes tell you that's 34% of the nasal zone that's covered with stain? I think as we get more and more specific like this, we need tech. And,
Yeah.
of course, I want to say AI because that's the buzzword these days. I don't think it is AI. I think it's actually machine learning and
Yeah.
basically educating your computer on counting pixels and assigning dots. And we're going to get there. We're not there. There are several people working on it. Lexitas have done some really pioneering work, I think, in trying to objectify an area that's unfortunately fuzzier than anyone wants to admit.
Yeah. There are some exciting things out there in some of even the phase 3 trials that are going on right now that will hopefully be a step in the right direction, particularly out of Boston. Now, you mentioned something that's really important, I think, which is that corneal fluorescein staining is not a direct indicator of active inflammation on the ocular surface. Now, probably most dry eye medicines in development have some sort of modulation of the immune system, right? Some sort of anti-inflammatory component. So, how do you typically go out to pick out the patients that are most likely actively inflamed?
Um, that's a really hard question. So
Yeah.
let's think about our diagnostics in dry eye. All right. There was, dry eyes changed so much. When um, inflammation was introduced as a concept, and that really goes back probably to 2000. Of course, I'm citing history books because I was still in high school then. Um, Allergan spent 3 years educating everyone on inflammation being a driver in dry eye. Absolutely a world-changing point of view, and excellent work, really well done. Animal studies first, human studies later, proof positive, indisputable. Let's go there. So then you have many manufacturers coming in with all kinds of anti-inflammatories. It could be T cell. There are many other mechanisms, and that is one of the exciting areas where dry eye research is still going, is other arms of inflammation, some that I'll even say most clinicians have never heard of. Who knows about aldehydes? I'll stop there because I don't
Mhm.
want to go too deep, but not enough people. And the same way no one thought about a T cell before, we're going to be talking about aldehydes. So
Yeah.
yes, there's a lot of development on anti-inflammatories, not exclusively so. I think there's a lot of other modes of interfering with dry eye that are relevant. But you asked, what's the best population? You can't answer that unless you know what your drug does. I
Yeah.
mean, is there a basis for saying my drug is going to work better in severe patients? If you know that, then you can select for a moderate to severe group as you design your trial. But very often that's not known. And I think it's a little bit of guesswork from very bright scientific officers in sponsor companies. And I'm forever impressed with the level of knowledge outside of the clinician group of scientists. And I mean, people who work in companies are downright brilliant. I've been impressed with
Mhm.
that in the beginning, and that's really never changed. Your mode of action may define the group of patients you want to look at. I think there's a leap of faith involved very often, but you might say, oh, I need the more severe group. More likely, I think you might say, no, I need the aqueous deficient group as opposed to the evaporative dry eye patient group. That's a lovely wish, except most dry eye patients have evaporative dry eye, at least 70%. And I think if you knew that, for example, if you had a drug that only increased aqueous production, you know that's what you want to interfere with. Maybe it's 10% of the clinically diagnosed dry eye. And being able to exclude patients with very variable degrees of evaporative dry eye is a gigantic challenge because we don't have good ways of scoring that at all. You asked, how do you define inflammation? Well, we've got an MMP9 test for tears, but it's too basic. Right? It's a
Yeah.
yes no test. It didn't need to be. It's the way that company chose to get it approved. They're able to quantitate how high the MMP9 level is, but that's not what's in the commercially available product. That's a big gap in our ability to be specific about how inflamed a patient is. Example, you may use a therapeutic that reduces inflammation by 75%, but your test is still going to be recorded as positive, even though you brought your MMP9 level from 300 down to 50, which is clinically maybe very, very significant, but it won't show up in your test. So we don't have, if you want to call it point of care or clinic-based diagnostics to define level of inflammation. People think they do it all the time, but they don't. Osmolarity, very interesting. Maybe there's still an educational gap there. You know, I know a lot of clinicians who don't use it. They say, oh, that's too variable. I don't know what it means. Well, there's pretty good science, and I credit Mike Lemp for doing a lot of work on this, saying that the variability, for example, between eyes is the earliest way to diagnose dry eye. So you might diagnose it with a threshold number, which has changed, was 317 when it got started, now 308, and now a difference of 8 between eyes. Any of those may diagnose dry eye. I think, and it may relate to company size, more education needs to be done to get people to understand that and also to believe it, because variability between eyes is not a problem. The other reason people don't use it a lot is reimbursement. There are still some major carriers that don't cover it. And again, speaking as a real world clinician, if 20% of your patients wind up getting the billing denied, you're not so eager to keep buying cards and keep doing a test. And I know a lot of people who stop doing the test just for that practical reason. Clinicians do
Yeah.
like to get paid for what they do. So I'll stop talking because I can just keep going on any one of these topics. I'll
Yeah.
let you refine the questions.
Yeah, no, well, no, so one of the things you hit on, is something that you've educated me on, which I think is really poignant, which is in evaporative dry eye. A lot of people are going after evaporative dry eye, but we sometimes see discordance in the eye dryness and symptomatology scales that we go after, eye pain or dryness or however we phrase it. And you've talked to me about evaporative dry and the importance of ensuring that the symptomatology is from the ocular surface and not from the eyelid or from others. Can you talk a little bit about confounding in symptomatology in dry eye?
Sure, and it gets considerably more complicated than the way you
Yeah.
just said it. So
Yeah.
it's this is probably not what I ought to say, but as much as we have learned about dry eye in the last 20 years, from my point of view, we know nothing. And we're terrible at treating this disease, absolutely terrible at diagnosing it and at treating it. Let's go to the evaporative dry eye or MGD. I think everyone would believe that 70% of patients with dry eye complaints have abnormal lipid in their tears. Well, how do we know this? Can we measure lipid layer thickness? Not very well. Can we describe the families of lipids that are in the tears? Not very well. Can we look into the glands and say, are they functional or not? A little. That's new that we have mybography and quite a few tools to do that. But I'm going to share some personal things that they're really not where they need to be. I draw as my case in point. I do a lot of meibomian gland probing. I'm taking a little, for those who don't know it, I take a little rod and pop open obstructions in meibomian glands. So I may look at a patient who's got 5 visible stripes on their mybography, but when I take them to probing, somehow I can get into 23 glands. My point is the absence of a white stripe on mybography does not mean you have a dead or atrophic gland. It does not. That's a problem. That's a real problem for the companies developing myographers. They don't want that word to get out because they may not sell as many machines, but that's the reality. And I don't know what a white stripe means. I don't know if that means normal meibum as opposed to abnormal meibum. Maybe we need a different wavelength. We need to better be able to understand, are meibomian glands functioning or are they not? Point number one. Um, that's a major problem in evaporative dry eye. So trying to define the population. Now you asked, well, where does inflammation come from? Another feeling I have, and this really comes from having done a fair amount of probing. Most people think of evaporative dry eye as a problem because you have insufficient lipid in the tears. You get evaporation of tears, you get nerve exposure, you have symptoms because of nerve exposure. Very, very logical. I call that lipid deficient evaporative dry eye. But I know I've got patients who have something different, and I call it oil retentive dry eye. I explain it to them as you've got these little capped off glands, and you essentially have little abscesses in your lids. And if I don't let it out, your symptoms don't go away. So that's a very different mode of symptoms and of inflammation by the buildup of that old and stagnant and now inflammatory oil within the lid. So I think some symptoms and some inflammation is lid origin, and some is abnormal tear origin. That's there are lumpers and splitters, and obviously I'm a micro splitter, but I think that's a differentiation that is important clinically and has yet to be looked at all in any kind of clinical trial.
You- Joe, you mentioned the corneal nerves being exposed. Can you talk a little bit about whether or not we should be testing uh, for neurotrophic corneas early in dry eye disease? Like, at what point does the dry eye cornea turn into like a neurotrophic type cornea?
I didn't know we were going to do the impossible questions today. Of course we should. Of course we should. But how should we do that? You know, what we talk about the discordance in symptoms and signs in dry eye, and I'm a crossword puzzle guy, and we know if you get to the end of the puzzle and there's still 6 squares, you did something wrong 9 steps ago. And that's really what's wrong with dry eye. We're making some incorrect assumptions, and that's why we're going wrong. I think every patient has multiple pathogenic processes that are the problem. There are patients who don't make enough water, obviously. There are patients with abnormal lipid, obviously. Nerves get, I'm going to speak generically, get retrained to
Mhm.
communicate a message of discomfort, even when the physiologic abnormality may have been corrected. It's what we're calling neuropathic pain. And I think that's in a lot of patients. Now, clinicians go through the way we know how to do things. You exclude the things you can treat first. If they're aqueous deficient, you replace it. If they're oil abnormal, I'm going to say, you do the best you can to normalize that. Maybe it's with all the lid hygiene, the squeezing, the probing. We still don't have replacement meibum available, which would be wonderful. But there are
Mhm.
some patients who don't have such abnormal water production, who have not clinically abnormal glands, and they're still miserable. And those are the ones we're labeling neuropathic pain. And there are some strategies to go further in that. Well, how do you diagnose that? Well, you can take out your confocal microscope and look at their subbasal nerve plexus, but how many people have that instrument and how many people know how to do it? And it's a 30 to 40-minute exam, so that's not going to happen.
Mm.
The problem is that virtually all dry eye patients have some component of this, and clinicians are just getting around to understanding this is common. This is not 1 or 2% of the dry eye patients. Clinical trials, I have no idea how we're going to incorporate that.
Mhm.
No idea. Um, we'll see. There are some real experts in this country um, who are good friends, and I speak with them all the time. There's a lot of work going on in this space. And by the way, on the clinical trial side, there are a number of companies looking at products that may intervene in this way.
Mhm.
Products that affect the corneal nerve receptors. I'm not going to name them because there are too many companies and I don't want to leave one out, but there's a lot of work going on to try to normalize or alter at least the perception that we call dry eye of abnormal tear coverage of the corneal surface.
Yeah, absolutely. And I know that the work that was done with the growth factors really just set off everybody thinking about that because of how successful those were. So it's it's an interesting area. So we only have a couple minutes left, but Joe, this has been fantastic, and you've been helping me with dry eye development for a decade now, and you've been helping Lexitas for just as long. So we're so happy that you've joined the team, and it's already making a big difference in our in the quality of the design of trials that we're doing. And it's I hope we can continue to innovate and get this right. Any questions from any of the webinar attendees at this point for Joe?
But while those come in, I'll just fill up your dead air time and talk about what I see as a little bit of future. I think
Yeah.
technology is going to get deeper and deeper into clinical trials. We spend a lot of time trying to find good study coordinators, better training programs to get more coordinators, to get more research sites. The transference of information from charts into clinical trial records. I think we're going to get much, much better on electronic source documents, things that live in the cloud, speeding up collection of data, reducing how many monitoring visits are necessary in studies.
Mhm.
The world of clinical research is changing and rather quickly.
It's beautiful. Yeah, absolutely. It's a fun time. Well, I'd like to thank everybody for attending, and I particularly like to thank Joe for sharing with us today. This has been fantastic, and we're looking forward to sort of the next generation of dry eye trial design.
Well, thank you for giving me a chance to spout off. And obviously, I'm very excited about what we're doing.
Yes, absolutely. All right. Thank you, everybody. Have a great day.
Thank you, everyone.